I'm really pleased today that our host is Professor Andy LiWang, who is a colleague here at UC Merced and one of our principal users in the NMR facility. And also that we're getting into the area of bio-NMR, which I hope that we'll do more of. So let me hand things over to John.
Thank you, Dave, and good morning, everyone. Good afternoon, good evening, wherever you may be. And welcome to our second Ivan meeting for June of this year. Had one a couple of weeks ago. Anyways, today we've got two very, very good speakers and very good subject material, very interesting subject material, as always. I always like to mention, please consider doing business with the smaller companies in the NMR industry, such as MR Resources and Q1 Instruments. I guarantee you'll be glad you did. Anyways, I just wanted to mention one other thing. Our growing video library of Ivan meetings on the MR Resources YouTube channel. Also, you can see them on the Ivan website as well. I think nearly 60 videos at this point of previous meetings. Have a look, and they're really been growing in popularity.
So let me just go right ahead and introduce Professor Andy LiWang, and he's going to get started.
Thank you, Dave. Thank you, organizers. I'm at the University of California, Merced, and I'll be talking about this protein Chi-B, and it's part of the circadian clock of cyanobacteria, and we're going to be looking at its temperature-dependent behavior and the potential role it plays in the temperature compensation mechanism of this clock. So all clocks, even mechanical clocks, are temperature compensated in that, if it's a cold day or a warm day, the clock still is able to tell time accurately. But the mechanisms by which circadian clocks are able to compensate for changes in temperature while maintaining the same 24-hour period is still kind of mysterious. And so this is one of the hallmarks of circadian clocks. And so I'm really interested in how clocks are able to do this. Then after my talk, John Orban will give a talk. And he's at the University of Maryland, and he's been there for 34 years. But right before he left, John and I shared this. We were in the same lab. In fact, we shared an office, and this is the lab of Brian Reed at the University of Washington in Seattle. And so we've known each other for a long time, and here we are co-authoring a paper when instead of studying proteins, we were studying nucleic acids. And this was back in 1992. Okay, so yeah, and just let me say, and John has been doing fantastic work on designing proteins that can switch folds. And so I really am looking forward to hearing his talk. All right, so to circadian clocks, you find these clocks in diverse organisms. And they regulate your metabolism in anticipation of sunrise and sunset. And they've been shown to be really important for health. You find them in mammals, plants, insects, amphibians, fish, even fungi, and then cyanobacteria. Now, I'm really interested in biological timing, but eukaryotic clocks are complicated. The clock proteins are, typically over 1,000 residues long. They're really difficult to purify. You have to, express them usually in these eukaryotic expression systems. So we decided to look at the clock of cyanobacteria, and they have the simplest known clock. And their proteins are relatively small. They're easy to purify. And so we decided, okay, let's look at the cyanobacterial circadian clock. And in 1998, Masahiro Ishira, Susan Golden, Carl Johnson, and Tako Kondo they published their discovery of the core clock genes of cyanobacteria, and they're called Chi-A, Chi-B, and Chi-C. And what's interesting is if you mix recombinant forms of Chi-A, Chi-B, and Chi-C with some ATP, you can the clock actually will work in vitro. And here is just from our lab, we just labeled one of the proteins with a fluorophore, and then we can just watch the fluorescence signal for a really long time. And you can manipulate the clock, you can perturb it with, temperature or metabolites. You can change the ATP, ADP levels. So it's a really powerful system that you can study in real time. And even in like NMR, you can, like, this is just a 1D. You can see that the clock oscillates in the NMR spectrometer, and here you can see that you can even look at it by 2D NMR, and so it's a really interesting system for biochemists, and so let me just give you a little overview of the clock so that you kind of know how it works. So every morning, Chi-A, which is a homodimer shown here in green, binds to Chi-C, which is a homohexamer, and each protomer of Chi-C has two domains. It has a light blue domain down here and a darker blue domain up here, and it has two residues, a serine and a threonine, depicted as these gray circles. And in the morning, they are not phosphorylated, but interaction of Chi-A with Chi-C stimulates an autokinase activity in Chi-C, and so that by midday, the threonine here at position 432 becomes autophosphorylated, and so here you can see that's depicted in red, and then continued stimulation of Chi-A, then Chi-C will become completely phosphorylated, where now the serine is also phosphorylated. Once a serine is phosphorylated, there's a conformational change that's transmitted down from these blue domains down to these lighter blue domains, and that allows this kinase SASA to bind, which helps this protein Chi-B that is the center of this talk to bind. And SASA is a kinase, and it stimulates this transcription factor RPA by phosphorylating it, and then RPA binds to promoters, and then that initiates expression of clock-controlled genes. Now, as we enter evening, more Chi-Bs bind to Chi-C, displacing SASA, and then recruiting Chi-A down here, where Chi-A is captured in an inactive conformation so that it can no longer stimulate Chi-C to autophosphorylate. So now this nighttime complex allows Chi-C to start dephosphorylating. And first, it dephosphorylates on the threonine residues. This nighttime complex also recruits KIK-A. KIK-A dephosphorylates RPAA, and that shuts off gene expression. And you can see this temporally separation of activation and deactivation of RPA creates these circadian rhythms of gene expression. And as the night progresses toward dawn, eventually the serine also dephosphorylates. And once it dephosphorylates, the Chi-C undergoes a conformational change back to the daytime state so that Chi-B down here is released, and that frees KIK-A and Chi-A so that Chi-A can start a fresh round of Chi-C autophosphorylation the next morning. So this is basically how the clock works, and we're going to focus on Chi-B. Notice how Chi-B only binds at night. It also requires it actually has two different folds, which is really interesting, and only one of the folds binds to Chi-C. So here are the two different folds of Chi-B. There's an inactive fold, which cannot bind Chi-C, and then there's an active fold, which can bind Chi-C. And to highlight some of the changes in the structures so that you can see that they have these two different folds, if you look at this terminal beta strand labeled beta 4, you can see in the inactive fold, it becomes this alpha 3 helix in the active fold of Chi-B. And then you see here you have this alpha 2, alpha 3, this pair of helices in an inactive fold of Chi-B becomes these beta strands, beta 3, beta 4, in the active state of Chi-B. And then you have these 3 proline residues that are all trans in the inactive fold. They have to go to a cis in the active fold. So we want to study how this protein goes back and forth, but the inactive in wild type Chi-B, the problem is that the inactive fold is so stable that you can't see the active state. It's basically invisible. And so it makes it a little challenging to study these transitions. So what we did was we made some mutations in Chi-B such that at room temperature, the active and inactive folds of Chi-B are approximately equally populated. So for example, here's an HSQC of that mutant at room temperature. And you can see that here, the residue A8, it has 2 peaks in this HSQC. One peak belongs to the inactive fold. The other peak arises from the active fold. Just a couple more examples. E35, you see 2 peaks. G89, there's 2 peaks. K43, V14, G78. And so by integrating these peaks, then you can get the populations of these 2 folds. And so we were interested in the temperature-dependent behavior of this mutant of Chi-B. And a former postdoc in my lab, Ning Zhang, so for instance, when he was studying this protein, he puts this Chi-B at 4 degrees, so like in the refrigerator or in a bucket of ice, and then he sets the NMR spectrometer to 35 degrees, and then drops the NMR sample into the spectrometer and starts collecting HSQC spectra. And initially, the peaks that he sees are predominantly from the active fold of Chi-B. But as time goes on, you can see that there are peaks starting to come up that belong to the inactive fold of Chi-B, and the peaks that belong to the active fold of Chi-B start to get weaker and weaker, whereas the peaks from the inactive fold of Chi-B get stronger and stronger. And he also measured, looked at the relative populations of this construct of Chi-B under equilibrium conditions at these different temperatures, 20, 25, 30, and 35. And so he could use those populations to calculate the change in free energy in going from the inactive to active fold. And he finds that as the temperature increases, the inactive fold becomes more stable relative to the active fold.
Want to mention our sponsors, as always, as well, MR Resources. Been around for nearly 40 years at this point, and offering virtually any and every sort of repair service, maintenance service that you might need for your Brooker systems, any Brooker systems, Avance 3s, Avance 3 HDs, Neos, Ultra Shield Plus magnets, Ascend magnets, on and on and on. They're equipped to handle it, quench recovery, system relocations, repairs. They've got also a very, very well-equipped probe shop. Can do, once again, literally anything and everything with cryo probes and cryo platforms. Do check them out if you would.
And he also measured the kinetics of fold switching by Chi-B, this construct of Chi-B, using this temperature jump approach. So he incubated the Chi-B sample at 4 degrees. He set the spectrometer at 20 degrees, and then he drops the sample in and collects spectra. And then he can then measure the kinetics. And you can see here the population of the active fold of Chi-B decreases over time, and the population of the inactive fold of Chi-B, the inactive state of Chi-B, increases over time. And then he repeats this by jumping to higher and higher temperatures. So here he's going from 4 degrees to 25 degrees, and you can see the kinetics of fold switching are increasing. And then he jumps the sample from 4 to 30 degrees, and then from 4 to 35 degrees, and you can see that the fold switching kinetics are increasing. And by fitting this data, he can then get he can estimate the free energy barrier. And in this case, going from the inactive to active fold, he sees that this free energy barrier is getting higher and higher with temperature. But the reverse reaction, going from the active fold to inactive fold, the free energy barrier is getting lower and lower. And so you can also look at this a different way, which is you can look at the Q10 values, which is you're looking at the rate of a process, a ratio of rates of a process where you're dividing the rate at one temperature by the rate at a temperature that's 10 degrees lower. And so here you can see that the Q10 is around, for going from the inactive to active fold, is around 2. But going from the active to inactive fold, the Q10 value is much higher. And so you can take all of this information, kind of make a cartoon of the energy landscape of Chi-B as a function of temperature. And so you can see that as the temperature gets higher, going from the inactive to active fold, this free energy barrier gets higher and higher. Whereas going from the active to inactive fold, you can see that the barrier, this free energy barrier, gets lower and lower for that transition. And so the question we had is, well, could this play a role in the mechanism of temperature compensation in the cyanobacterial circadian clock? So we did just a really simple modeling where you have here you have the rates of going between the active and inactive folds of Chi-B. The active fold of Chi-B can bind this domain of Chi-C to form a complex with on and off rates. And so we modeled this, and we first modeled this assuming that the free energy landscape does not depend on temperature. And what we see is that that in this case, at these different temperatures, you can see that the amount of Chi-BC complex formed over time is quite different. But then what we did is we then remodeled this binding of Chi-BC, but using our experimentally determined free energies and free energy barriers. And here we see that that the amount of Chi-BC complex formed is much more similar at different temperatures. So we think that the temperature-dependent behavior of Chi-B does play a role in the overall mechanism of temperature compensation in this clock. Now, the other thing that's interesting is that Chi-B takes hours to bind Chi-C. And here is a gel mobility shift assay run by Gary Chow, a former graduate student in one of my collaborators' labs, David Britt at UC Davis. And here you can see that when he mixes Chi-B and Chi-C together, the complex, the amount of complex formed it takes hours to form. And so this is important for the clock. You want things to happen very slowly because the period of the clock is 24 hours. So what's great about studying a circadian clock is things happen really slow. So you don't need any kind of fast instrumentation. So we thought, well, gosh, could the slowness of binding be because the Chi-B has to go from the inactive state to the active state, and these 3 prolines that are trans have to become cis? Could that be the reason? So to test that idea, a former postdoc in my lab, Damini Sood, she was looking at the rate of fold switching of Chi-B under a temperature jump from 40 to 35 degrees with increasing concentrations of a peptidyl prolyl isomerase, which catalyzes prolyl isomerization between cis and trans. So here she has no isomerase, and then so you can see the kinetics of fold switching here. And then she added 10 micromolar of this isomerase, and you can see that the fold switching increases, and then the rate of fold switching increases. And then when she adds 50 micromolar of this isomerase, you can see that the rate of fold switching increases even more. And so there's this dose-dependent behavior of fold switching of Chi-B with concentration of this isomerase. And so this suggests that indeed the rate determining step in Chi-B fold switching is this prolyl isomerization. Now, we also use hydrogen deuterium exchange NMR, and what we found is that this region with the 3 prolines undergoes fairly fast exchange, much faster than the rest of the protein, and using basically mild denaturants like urea, and looking at the rate of exchange as a function of urea, we were able to determine that this region is quite flexible, quite dynamic, and we think that having this localized unfolding of this region of Chi-B that encompasses the 3 prolines is important to allow these prolines the freedom to isomerize. Okay, so that concludes my talk, and I'd like to acknowledge Dominique Su, Ning Zhang, Gary Chow, and David Britt, Susan Golden, and Carrie Parch for collaborating with me, the Army and the NIH for funding this work, and UC Merced. And thank you very much.
Good afternoon. Q1 is dedicated to developing NMR hardware and software to make routine NMR more accessible to a broad range of scientists and engineers. The company aims to establish new and innovative products for demanding applications across the sciences. Our products consist of complete systems at 400 megahertz with magnets manufactured by Q1. We also offer complete systems with magnets from MR resources up to 600 megahertz. Our console upgrades in 2 or 3 channels from 300 to 600 megahertz, with the economy console consisting of the Quantum 1 Plus console, preamplifier, workstation, and utilizes the existing magnet probe and shims. This is by far the most economical upgrade in the market. A full upgrade consists of the Quantum 1 Plus console, preamplifier, broadband STM gradient probe, and sample changer for a completely automated system at a price that would surprise you. And our smart tune and match probes for Brooker invariant systems for improved throughput and reliability. Q1 probes are fully integrated into the Topspin and VNMRJ platforms. The Q-Link Ethernet-based interface can be installed on a wide range of consoles to fully automate the operation of older NMR systems and add multi-nuclear capability to your dual or quad nucleus spectrometer. Want to know more about our probes, consoles, and complete systems? Contact us for more information or a remote demonstration. Thank you.
So I'd like to introduce my friend, John Orban.
Thank you, Andy. That was a great introduction to my talk. I'm going to be talking about designed folds. Okay, so my lab is interested in proteins that can populate more than one folded state. And I'm just going to show here a diagram that summarizes a sort of a continuum of different levels of stability and flexibility in proteins. And on the right, the bottom right here, we've got proteins that are very stable, that have deep energy wells. And on the left-hand side up here, we've got intrinsically disordered proteins that are highly flexible, and they have very poorly defined energy wells. And both of these types of proteins have been studied extensively. But what is significantly less studied are these types of proteins here that can switch between 2 different ordered states, the sort that Andy has just described. And these sorts of proteins tend to have shallower energy wells and which allows them to get access to alternative states and populate them. And it's these types of proteins that I'm going to talk about. So, this slide just shows some naturally occurring protein fold switches, one of which is Andy's here that he has just given this beautiful presentation on. But there are others as well. And to define fold switches, you could look at one of these structures, and the sorts of things that are features of protein fold switches is that there's one region within the structure that undergoes fairly large amplitude structural changes where there's very big shifts in the secondary structures. So for example, here in RFAH, this helical hairpin undergoes this large conformational change into a beta barrel, for example. And so these are proteins that are 100% sequence identical to each other. There's no mutational change, but what is causing the change in conformation is some sort of environmental factor. And that environmental factor can be ligand or pH or redox. Most commonly, it seems to be ligand. And to date, there are about a dozen of these that have been naturally occurring switches that have been really quite well characterized. But a few years ago, there was a paper that was published from Lauren Porter and Luger that estimated, there was a computational analysis of proteins in the PDB that estimated that there are actually considerably more proteins out there that can undergo this fold switching. So this prompts the question then of, well, can you predict and design protein fold switches? Now, we know that there's been significant advances in being able to predict and design unique protein structures. AlphaFold has been a wonderful demonstration of that. But if we really understood the protein folding code, we should be able to predict and design proteins that switch. And there has been some recent progress in predicting some naturally occurring fold switches, but the design of fold switches remains fairly challenging. And so why do we want to design fold switches? Well, so the most fundamental reason is that it would help us resolve some of the ambiguities in the protein folding code. This would lead to improved understanding of sequence structure relationships and would lead to the development of better prediction tools. And there would be downstream benefits from that I've listed here. But there may also be some practical benefits as well. So we've worked on protein fold switching for quite some time now, but what I want to do is talk about some of our more recent work where we've developed switches between three commonly occurring folds. And so this one here is 3-alpha, is a 3-alpha helical bundle that in our context binds to albumin. We call this GA, but it's also seen in many homeodomain proteins. There's this S6 fold, this alpha beta plate, which in our context is a ribosomal RNA binding protein, but is also seen in eukaryotic proteomes. Many RNA binding proteins have this fold. And this fold here, this beta grasp fold, which is a ubiquitin-like fold, but in our context is involved in IgG binding. So the question that we're really trying to answer here is, to what extent can you embed the sequence for one of these folds into another fold? And I'm going to focus mostly on this particular switch here. So really what I'm interested in is, to what extent can you encode this 3-alpha sequence into this alpha beta plate? And I'll just briefly summarize the design approach here. That the main idea is we want to thread the 3-alpha sequence through the S6 fold, and they're in multiple alignments. And we want to find an alignment where we minimize the worst steric clashes. And once we've found that alignment, we want to make mutations to relieve those clashes. That's the basic idea. And so the idea is then that we're going to try and co-evolve these sequences. So we're going to try and make the GA sequence more S6-like. We're going to make the S6 sequence more GA-like. We're trying to hybridize these sequences to ask the question, well, how high in identity can we go between these sequences and maintain the parental folds? And the surprising answer in this particular case is that we can actually transplant all of the 3 alpha sequence into the longer alpha beta plate fold. So here, for example, we have, so this is the GA sequence, all of the 56 amino acids. If we just look at that sequence on its own and solve the structure of it, it's a 3 helical bundle. When it's implanted into the longer sequence, all of it, 100% sequence identical over this 56 amino acid range, that corresponding sequence has a very different NMR spectrum. And when we solve the structure of that, it has this alpha beta plate fold. Now you can see that there's some commonalities here. This blue helix is the same between the two structures, but the red and yellow helices become strands or parts of strands in the alpha beta plate. Now, when you get to these really high sequence identities, you develop some very interesting properties, as Andy has shown here. So at this high sequence identity, you've basically developed a critical state where the two folds are in competition with each other. And so if you make mutations now, we typically think in protein folding of 2-state systems where we either have a folded state or an unfolded state. But in this particular case, it really does depend on where you make the mutations, because some mutations can lead to unfolding. Other mutations can just lead to the alpha beta plate. And if you make mutations that destabilize the alpha beta plate, completely destabilize the alpha beta plate, you can unmask the alternative 3 alpha fold. So you have this 3-state system now, but if you make mutations that are more subtle than this particular one, then you can populate both states simultaneously. It's similar to what Andy showed in his talk. So in this particular case, you look at the NMR spectrum, you see 2 sets of signals, one due to the 3 alpha, which are the ones labeled in red, and the other due to the alpha beta plate. Now, this particular mutation is a mutation from valine to threonine, and it occurs on the periphery of the hydrophobic core of the alpha beta plate. So it destabilizes the alpha beta plate, but not completely. It destabilizes it enough that you can populate this 3 alpha fold in equilibrium with it. Now, this particular equilibrium has a temperature dependence to it. And so in this particular HSQC spectrum here, if I just expand this region here, you can see these glycines, glycine 58 and glycine 34. And at low temperature, you see just these red signals due to the 3 alpha state. But as you increase the temperature, you start to see these new signals growing in, which are due to the alpha beta plate, these blue signals here. So by 30 degrees, it's mostly alpha beta plate. So we've actually solved structures at the low temperature and high temperature. And low temperature is a 3 helical bundle. At high temperature is an alpha beta plate. And this is reversible. We can go back and forth between these, and these are monomeric systems. An interesting feature is that this system is very sensitive to mutation. So we can shift the temperature midpoint by making relatively subtle mutations at the periphery of the hydrophobic core. So for example, on the left-hand panel here, we've got an isoleucine here, and you can, under this condition, you can barely detect the fold switch. But if you mutate the isoleucine to a leucine, you can now readily detect the fold switch even at low temperature. And so fairly small changes in hydrophobicity and branching on the periphery of the core are leading to these changes. So the system is quite delicately balanced, which is consistent with it being in a critical state. Because we've got temperature-dependent data, we can integrate these peaks and get equilibrium constants and then get free energies for the conversion from 3 alpha to alpha beta. And so this energy conversion, this free energy going from about more than 90% 3 alpha to more than 90% alpha beta over this temperature range, it takes about 2 kcals per mole to do this. So these systems, these two folds are relatively close in stability. And you can recast this a different way and plot it in a Van't Hoff format. So the negative slope of the Van't Hoff fit, where you plot the log of the equilibrium constant versus the reciprocal of the temperature, gives you the enthalpy contribution to the reaction. So this is the negative slope indicates it's an endothermic reaction to the tune of heat absorption of around 20 kcals per mole. And we can also get information about the kinetics of the interconversion. So we use an experiment, the ZZ exchange experiment, which I'm sure many of you are familiar with, where you not only can see the backbone amide signal of in this case, the glycine 58 in the 3-alpha state and the alpha-beta plate form, but also the exchanging peaks between the two states. And you can measure the peak intensity of these peaks as a function of time to get kinetics for the forward and reverse reactions. And you can see that the fold switching event is slow on the NMR timescale. It's on the order of about a second. And so that's consistent with the fact that we can see both sets of signals. But the most important thing here is that this particular exchange experiment shows that these two systems, the 3-alpha and the alpha-beta plate, are in fact correlated. They're not independently varying 3-alpha and alpha-betas. They are actually in exchange with each other. So we did this experiment at 15 degrees, but you can do it at different temperatures, of course, and that enables you to do an Arrhenius analysis where you can plot the log of the switching rate versus the inverse of the temperature and get information about the energy of activation. So the energy of activation is around 22 kcals per mole, which suggests that the 3-alpha to alpha-beta reaction is dominated by an enthalpic barrier. Now, interestingly, the reverse reaction has a much smaller energy of activation, and we don't fully understand the reasons for this at this moment. But one thing that we notice is that when you go from alpha-beta to 3-alpha, you're actually unfolding about 40% of the polypeptide chain as you lower the temperature. So energetically, this is a little bit like cold denaturation. And cold denaturation we know is an exothermic process. So one possibility is that the putative heat release from the exothermic process is offsetting the energy requirement for this energy of activation from alpha-beta to 3-alpha. And that's our working hypothesis for now as to why there is an asymmetry in these energies of activation. So how is it that you can code for two different folds with a single sequence? We've developed these 3 switches, and I just want to go over the common features of these 3 switches just briefly. And the first is this idea that we have minimally overlapping cores between the switches. So if we look at the top right structure and you look at the core residues, I've highlighted those in the beta grasp. And if I map them onto the alternative state, they map to solvent exposed positions. Okay, and so we think that helps to encode the 2 different folds into a single polypeptide chain. So now if I go to the next slide. Can you see the next slide?
Yes.
Okay,
Yes.
Thank you. So this highlights the fact that disordered regions play a role in remodeling the ordered states, and basically you can see that disordered regions can become ordered in one of the states, and sometimes they're at the ends of these folded regions, and sometimes they're disordered loops in the center of the polypeptide sequence. Let's see, so I think we'll catch up here. So now you can see the next slide. This is looking at overlapping stability profiles.
Yes, we can see.
Okay, great. So, all right, seems like we're on track now. So this plot shows the stability profile for the 3 helical bundle is in blue, and the alpha beta plate is in black, and you can see over this temperature range they don't overlap. But when you make this mutation, this subtle mutation from valine to threonine, it now makes the stability profiles overlap. Of course, overlapping stability profiles are a necessary requirement for fold switching, but it enables the stability. You can see quite clearly the stability profiles are overlapped. Now, mutations can do this, but also environmental factors can do this. And all you really need is an equilibrium amount of the alternative state, and it can be weakly populated. But if you can, as long as you have an environmental trigger where the energy gain from the triggering event can outweigh the energetic cost of the fold switch, you can also bridge this gap. And I have a couple of examples that I want to show from our design work to illustrate this. And so to illustrate this, I'm going to use the temperature-dependent fold switch that I just discussed. So at low temperature, the equilibrium for this reaction is far over to the 3 alpha state, but there is an equilibrium amount of the alpha beta plate present. And so if you design cysteine residues into the system so that they're far apart in one of the folds, but in close proximity in the other fold, then you can use redox exclusively to switch between these two states. So now if you use under oxidizing conditions, the free energy gain from disulfide formation, which is in the tens of kcals per mole, is outweighing the energetic cost of fold switching, which we know is only about 2 kcals per mole. Now, if you put the system back into a reducing environment, you can shift the equilibrium back to mostly 3 alpha. So this is an example of a redox triggered fold switch. There are naturally occurring examples of this as well. You're all seeing my slides change, right?
Yes.
Yes.
Okay, good. So, and the last example I want to give is one of ligand binding, how ligands can induce fold changes. And this is perhaps the most common case in naturally occurring switches. And this is for the switch between 3 alpha and beta grasp that we've developed. And each of these folds has its own cognate ligand. So 3 alpha fold binds its native binding partner is albumin. The beta grasp fold, its native binding partner is IgG. But when these proteins were designed, we included in them the binding epitopes for both ligands in all of the sequences. So this opened up the possibility of potentially having proteins that could be bifunctional. So we have a system like this where the 3 alpha state can bind to human serum albumin. It has a 3 helical bundle. But some of our mutants, high identity mutants, can populate the alternative state. So these intense peaks here are due to the 3 alpha fold, but you can see there's a weak set of signals here due to the beta grasp, the alternative fold. And we hypothesized that, well, we should be able to add the cognate ligand for the beta grasp, and it would shift the equilibrium over to the beta grasp because folding and binding are thermodynamically linked. So in fact, when we add IgG, which is the cognate ligand for the beta grasp, the minor signals from this particular spectrum become the major signals. And so the 3 alpha has shifted over to the beta grasp in the presence of IgG, and the free energy of binding outweighs the energetic cost of switching. So just to summarize, we think of fold switching systems like this, where you have a ground state and a higher energy alternative state, and that might be quite weakly populated, and you can shift the equilibrium between these states using mutations, and you can do that in multiple ways. In this particular system, we can make mutations at different points in the sequence and shift from one state to the other state and back to the other state and so on. And so you can shift with mutations or you can shift with environmental triggers. So just in conclusion, I hope I've showed you that we can design switches between very common folds. There are some recurring features that are present, and you can switch between these different states using either very short mutational paths like a single amino acid or environmental factors. And basically fold switching is a way to expand the functional repertoire of a given polypeptide chain, sort of like post-translational modification. So in the future, I think fold switches are going to help improve our understanding of sequence structure relationships, and that is going to lead to better prediction tools. But not just, I don't think it's going to be useful for that also. I think it's also going to be useful as a new strategy in protein design. At least that's our hope, and that's an area that we've spent quite a bit of time and continue to work in. I just want to acknowledge here my collaborator, Phil Bryan. We've been working on this for a long time, and most of the work that I described, switching between the 3-alpha and alpha-beta plate proteins, was done by a very bright graduate student in my lab, Saga Solomon. So thanks very much. I'm happy to take any questions.
That was a great talk, John.
Yeah.
So I have a question. So would you say that which fold has the more hydrophobic context, the 3-alpha or the alpha-beta plate?
Alpha-beta plate has a bigger hydrophobic core. So its hydrophobic core is about 3,200 square angstroms. Compared with the 3-alpha is about 1,700. And so, you know, we wonder, well, why does it do what it does, right? And so we know that the hydrophobic effect is temperature dependent. And we know that as you lower the temperature, hydrophobic effect weakens. Mm-hmm. And so perhaps one thing is because there's more hydrophobic effect in the alpha-beta plate, the temperature effect is greater for that than it is for the 3-alpha, and that it's enough of a difference that it causes this overlap of the stability profiles. That's, you know, that's a little hand wavy, I understand, but that's what we think might be going on.
Yeah, because in Kai-B, when we looked at the hydrophobic core between the two folds, the one that's more stable at higher temperature has more hydrophobic contacts than the one that's more stable at lower temperature. So
that's very consistent with what we're seeing.
Yeah, that's cool. That's cool.
And so I was interested in your cold, the lower, as you drop the temperature, you saw different effects also in the energetics, right?
That's right. So the energy barrier the free energy landscape is shifting.
Mm-hmm, mm-hmm. Is there a difference in the amount of disorder between your two states, or are they approximately equal? Like, do you have more disordered loops in the lower temperature state, for example?
I would say, yeah, I would say the lower temperature state is more dynamic.
Mm-hmm.
It's less stable, and in fact, if you just lower the temperature a little more, the whole thing unfolds. It undergoes denaturation.
Okay, all right. So this is actually there are parallels between your system and your natural system and our design system have a number of parallels. It's very interesting.
Yeah, yeah. Cool. Lower the temperature and well above freezing for our. Well, we did have to make mutations because the wild type Kai-B is so stable in the inactive fold that we had to kind of we had to shift it toward the active fold through mutagenesis.
Mm-hmm, mm-hmm.
And so we had to do a lot of looking at the structure like you do, because we had to design these mutations to shift this equilibrium.
Right.
But for that construct, yeah, you lower it and around 15 to 10 degrees Celsius, you can start to see it unfold.
Mm-hmm.
In fact, at 4 degrees, you know how we incubated Kai-B at 4 degrees and then dropped it into a spectrometer that was set like between 20 and 35 degrees? The Kai-B is completely unfolded at 4 degrees, but when we drop it in a spectrometer, it folds up. And so by the time you So by the time you take the first spectrum, it's folded, but it's adopted the active fold, and then it starts to shift toward the inactive fold over time.
That's very interesting.
Yeah, a lot of parallels.
Yeah, no, it's the biophysical principles, they're exactly the same.
Yeah, it's cool.
May I ask a question? Jump in.
Yes.
How do you decide for both you and Andy, on what mutations to make to stabilize your folds, and to what extent do you model all these conformations as you're going and deciding what to do?
So, we use Rosetta to energetically evaluate mutations.
Okay.
And so we model them before we make them, for sure. And that works most of the time. But, sometimes we get surprised, and so we have to go back. But yeah, I mean, we use computational tools to assist us.
What about AlphaFold 3? How does that compare to Rosetta?
So we've used mostly Rosetta. I don't think of AlphaFold as an energy calculator. It's not a free energy calculator. But I think it has some. I think AlphaFold, there are various iterations of it now that use subsampling methods that you're probably aware of, that may be able to predict alternative states, which I think is very exciting. We've used Dorothy Kern used our system and your system in her Nature paper, which is a beautiful paper, and I think had some success in predicting alternative states. I think some of our alternative states, AlphaFold cluster didn't do so well on. So I think there's certainly room for improvement. We haven't used it extensively yet in a design strategy, although that's something that we're very interested in doing.
What program do you use to thread a sequence through a structure?
Rosetta.
Rosetta.
Rosetta is a threading routine.
Okay. And then you make mutations, and then like if you want to stabilize the 3 alpha over the alpha beta plate, you put in a mutation, and then you use Rosetta to calculate the energetics of both folds, and then you see if it's shifting in the direction you want.
Right. And what we're trying to do is make something that will hopefully if it's a catastrophic effect, it's not catastrophic in both, or it can be relieved in one of them and have minimal effect in the other. So we're trying to accommodate both folds when we do those designs.
And when you use coevolution, what is your favorite way to do that?
The way we did it here was using threading computational approaches and then making mutations. Before that, we were using phage display, and that is technically can be quite challenging. And so we've moved, because the computational tools over the years, I mean, we've been playing with these fold switches for 20 years now. And so the computational tools have gotten a lot better over 20 years. So we've tended away from phage display and moved more towards using the computational tools.
What's your favorite computational tool for
well, still is Rosetta as an energy calculator. Yeah, I don't think AlphaFold is a very good. I don't think it. If you ask John Jumper, I think he would tell you AlphaFold is not a free energy calculator.
But for coevolution, like when you're looking at doing coevolution, isn't there? You need a computational resource to do the calculations for you, right?
Yeah, it's Rosetta.
Rosetta too. Okay,
yeah,
so
Rosetta.
Okay, I have to check that out.
But, you know, there's nothing beats the experiment, you know.
Of course. No, but like,
obviously, but that's our guide. That's our guide.
exactly. No,
And I'm hopeful, Andy. I'm hopeful one day that we will be able to use AlphaFold and AlphaFold cluster. I don't think I mean, AlphaFold for sure is a great predictor for unique folds, but it doesn't do so well with our switches. And AlphaFold cluster does a little bit better, but it's still, you know, that switch I showed at the end where the different mutations switching back and forth, it gets half of those right. So I think there's still room for improvement, and yeah.
So is Rosetta the Rosetta that you're using, is it a server that David Baker provides, or did you have to install it onto one of your own computers?
You can do either. You can do either.
Okay.
We should open it up to any other questions. We're kind of hogging it.
I'll say any other questions or other questions. Okay. Well, if there are no other questions, I certainly thank both of you for giving these talks. I'm fascinated by all that. I don't really have a full understanding of all the thermodynamics of protein folding, but it seems there's a little bit of everything in making these switches.
Yeah, and how important NMR is.
Yeah.
Right.
Yeah, I don't think we could have assessed what we were doing without NMR. It was absolutely vital.
Yeah. Well, thank you everyone for attending.
Okay. Well, thank you, Andy, and thank you, John. And if there are no more questions or comments, I will close the meeting.
And great to see you,
very much. Yeah, good to see you too, Andy. Okay. Take care.
Take care.
Thank you, everyone.
Bye.
Bye.
Bye, everyone.